Here, yeah, you can do it. Normally what I'm doing, normally what I've done is it's always that first piece to it. But normally, that's the time we look at the values really good.
Yeah, and I'm going to switch it over now. I'm going to follow you up just to see, and then I'm going to go into the booth and see if they're all good. Make sure they're all good there.
All right, why don't we go ahead and get started? Well, I'm John Carruthers. Actually, I'm not John Carruthers. But John's out of town, and I'm filling in, and I get to introduce today's speaker, which is a real treat for me. I've worked closely with Brian for the past few years. Brian's on our faculty here at the University of Michigan in hospital medicine, and he's a fantastic physician scientist who both does really terrific stuff in the wet lab, which you're going to hear something about, and is also a really great doc. I mean, so I think he's a fantastic role model of what a physician scientist should and can be, and it's a real treat to hear him talk here. Brian's an MD PhD, graduate from Northwestern, was a resident in our program here, and has been on the faculty attending in hospital medicine and working together with my group for the past couple of years, and in the process of setting up his own independent research program. And you're going to hear about a lot of that stuff today. So I think that's about, I'm not going to use up any other time. I think many of you know Brian, and I'll take the rest.
All right, well, thank you, David, for the introduction, and thank you all for coming, especially without there being lunch today. I appreciate it. I suspect that for many of you, when you see the name CRISPR, the word CRISPR in the title of this talk, the first thing that comes to mind are these kind of sensational news stories about how scientists have this master plan to make designer babies and to engineer the human race. Or maybe you've heard more about grandiose claims about how CRISPR is going to end hunger or pollution or all disease and change life as we know it. And what I'd like to do today is to provide you with a little bit of context and perspective about what CRISPR is and its history, and then to tell you a story about how we've been able to use CRISPR in the lab to make what we think are some fundamental insights into the molecular basis of cardiovascular disease. So you may be surprised to hear that CRISPR was first observed in nature over 30 years ago, back in 1987. And I think it's important to realize that this was not an applied question. It was not scientists setting out to find ways to change the human genome, but rather it was just scientists looking at DNA sequences in the E. coli genome, and they noticed these kind of funny regions of DNA where there were these regular interspaced short palindromic repeats, and that gave rise to the name CRISPR based on the acronym. So there were these regions of DNA that were regularly spaced by these intervening DNA sequences called spacer DNA, and this was just a curiosity. They didn't know what to make of this, and this was largely ignored for the next 15 years until more and more bacterial genomes began to be sequenced, and it was realized that these CRISPR arrays were not a curiosity or an idiosyncrasy of that original report, but that these were actually widely conserved DNA regions that were present in many different bacteria and archaea. And in addition to the CRISPR array, there were also associated genes that were homologous that were often found nearby, the CRISPR-associated or Cas genes. But still at this time, people didn't know why they were there, what they were doing. And surprisingly enough, some important insights came from the food industry. And it turns out that if you're trying to make yogurt at an industrial scale, it's important to understand how to culture lactobacilli and other fermenting organisms at a very large volume. And it turns out that one of the things that can go wrong when you're trying to do this is that just like we can get infected with viruses, so too can bacteria. And those viruses for bacteria are called bacteriophage or phage. And these can infect a bacterial culture and kill all the lactobacilli, and then you get no yogurt. So there were scientists working in this industry that were very much interested in why some bacterial strains were susceptible to phage infection and some were not. And they made two important insights. The first was that the DNA that was present in these spacer regions of the CRISPR array often matched, it was identical to the DNA that was present in the phage. It seemed to be foreign DNA from the phage that was acquired into the bacterial genome. That was observation one. Observation two was that the presence or absence of that CRISPR phage DNA closely correlated with resistance or susceptibility to phage infection. So if a strain spontaneously lost the CRISPR array, it would become susceptible to infection with that same phage, or if another strain acquired CRISPR DNA, it became resistant to infection with that phage. So with those observations in mind, many groups over the following 5 or 6 years worked out the mechanism for how this was happening. This is a complicated figure, but all you really need to understand is that the phage DNA gets acquired by the CRISPR system into the CRISPR array, and now the bacteria can encode for what we call guide RNAs, which I'll come back to in a bit. And the guide RNAs, together with the Cas enzymes, can now recognize phage DNA. If another phage comes along and infects that bacterial cell, it can now cleave that phage DNA, and it's resistant to infection. So essentially, this is an adaptive immune response. It's a memory that the bacterial cell has of a prior infection that renders it resistant to future infection. Once this machinery was worked out, it was realized that you could take that core machinery, the Cas enzymes and the CRISPR array, and you could transplant it. You could move it from one bacterial strain to another, or from one bacterial species to another, or you could even move it into virtually any cell type, including human cells. And now you could make changes to DNA. You could disrupt genes, or you could make precise changes in the genome of even human cells just using this CRISPR machinery. So it was a transferable means of changing DNA. And it was really this adaptation of CRISPR to human cells that provided us a tool to do genome editing and really led to an explosion of interest in CRISPR. And you can see, based on the number of publications in PubMed over the years that have CRISPR either in the title or the abstract, that around the 2012-2013 timeframe, there was really an explosion of interest in CRISPR. So how does this work? So if you want to change a target site of DNA in the genome, how do you do that? It's fairly straightforward. All you have to do is you have to get two things into the nucleus of the cell. One is the guide RNA. So this is what's encoded in nature by that CRISPR array. And this has, importantly, 20 nucleotides of sequence that scans the genome through billions and billions of base pairs of DNA and finds its perfect matching target site. The guide RNA then recruits a Cas enzyme. Most commonly, we use Cas9 from strep pyogenes. It recruits Cas9 to the target site, and then Cas9 is able to induce a double strand break in the DNA at that site. What happens next in mammalian cells depends on the repair pathway that's used. Mammalian cells don't like having double strand breaks in their genome, so they try to fix it. One way that they can do that is with non-homologous end joining, where they try to take the blunt ends and stitch them back together. But when they do this, they're not very good at it. They very often incorporate extra base pairs of DNA, or they delete base pairs that are supposed to be there. The result of this in a coding sequence is very often a frame shift and a complete loss of function of the gene. Alternatively, if there's a template available to the cell, and you can provide this experimentally, you can engineer a stretch of DNA that matches the target site that you're trying to repair, and you can incorporate specific nucleotide substitutions at that site. And now when the cell uses homology-directed repair, it will incorporate those desired mutations into the target site. So why is this so powerful? So this is a targeted, programmable approach. So rather than introducing random mutations in DNA, you can focus in on virtually any gene in the genome. It's a highly efficient process, unlike homologous recombination before it. We can get editing efficiencies that approach 100% in certain conditions. This also tends to be a highly specific reaction. So it's guided by that 20 base pair stretch of DNA, and you can choose target sites that are unique in the genome that don't have similar regions throughout the rest of the genome and try to get only changes at your desired site without off-target effects. And then finally, as we'll talk about a little bit more in a minute, this is a scalable approach. So because the programmability is in this 20 base pair sequence of DNA, we can synthesize guide RNAs at a massive scale. We can make tens of thousands or hundreds of thousands of guide RNAs. And also, when we read out their abundance or we measure them on the back end, we can use next-gen sequencing and get hundreds of millions of reads of guide RNA sequence. So naturally, as people began to do this in the lab and it became more and more routine to make changes in DNA, people naturally began to wonder, well, why can't we do this in patients? Whether you have a patient with sickle cell disease or cystic fibrosis, might you be able to go into the patient and use CRISPR to directly correct those disease-causing mutations, either by delivering the reagents in vivo into the patient or by removing from the patient either hematopoietic stem cells or iPS cells, engineering those cells, and then introducing them back into the patient. This is very exciting, and this would be paradigm shifting, that we could treat genetic disease at its molecular root, at the cause, and not just try to treat downstream consequences. However, there are a number of challenges that limit the application of this to becoming a direct therapeutic. One is that for this to be effective, you would have to reach high levels of delivery. You'd have to get the CRISPR reagents into enough cells. And once they're there, they would have to be effective enough. They would have to edit enough cells to get a therapeutic effect. And of course, we think about not just benefits, but also risks. If you had off-target mutations, you might get toxicity associated with that. And finally, I think what impacts all of those above considerations is the power of in vivo selection. And so David and I are in the process of writing a review about this as it applies to hematologic malignancy. And we have an illustrative example here where if you think of a hematologic malignancy that carries a driver mutation that makes it malignant, and for the sake of simplicity, let's just assume for a second that there's a single mutation and it's a driver mutation and it's present in all of the leukemic cells. Well, if you could use CRISPR to edit that causative mutation and you had 100% editing and you had no off-target effects, you would expect that you'd have a therapeutic effect. However, if you had incomplete editing, if you had even 1 out of 10,000 or 100,000 or 1 million cells that was not edited, that immediately might not be apparent. You might have a big reduction in disease burden, but over time, that leukemic clone would have a fitness advantage over the corrected cells, and you would expect that it would proliferate and lead to disease relapse. So you could argue that you might need even 100% editing efficiency. But even if you could achieve that, you have to worry about off-target effects, even if they are rare. So along the same lines, you might have an off-target effect that is irrelevant. It doesn't change the phenotype or the fitness of the cell. But if you have a mutation that maybe hits a tumor suppressor like P53, it could drive oncogenesis of that clone. And again, over time, that would have a fitness advantage and lead to disease relapse or a second malignancy. So what's important to take away from this is it's not just the editing efficiency and the off-target effect at the time of treatment, but also how that might change over time in a patient when there's a selection pressure. Just briefly, I want to say a word about germline genome editing because it's been in the news. So the idea here is that if you had a mutation that you wanted to fix in an embryo, that you might be able to do that. So in this paper, they looked at a disease model where one parent was heterozygous for a dominant mutation in myosin-binding protein C3 that causes a form of cardiomyopathy. And so the idea here would be that if you had embryos that were generated from these two parents, that some of them would be wild type, but some of them would be disease affected. And would it be possible, technically probably yes, to go in and to repair that disease-causing mutation so that you might have a repaired embryo that you could then transplant? There are risks of that you might get mosaicism or you might get off-target effects. Hard to know what might become of that, but the obvious consideration here, I think, is rather than trying to correct this embryo, why not just take this embryo? Half the embryos should be unaffected, and it's much simpler and much safer to just to implant that embryo instead. The same is true if you think of a recessive disease or a dominant disease. It's hard to imagine many scenarios where there's an unmet clinical need for germline editing. I think most people that think seriously about CRISPR as a therapeutic are not very excited about the potential for this kind of approach. But I think, regardless, this is still very exciting. There are barriers to somatic delivery of CRISPR reagents. There are a lot of smart people that are thinking about it and working on it. I think time will tell. But I think regardless of whether CRISPR is ever used in a human or not, in a patient, it's already had an enormous impact on our ability to do scientific discovery and to learn about the mechanisms of disease and to how to be better able to treat disease. So what CRISPR gives us is the ability to not just read sequences of genomes, not just observe natural variation, but to perturb genomes, to make precise changes throughout the genome and to investigate the consequences and to rapidly generate cellular and animal models of disease. And so I'd like to tell you a little bit about how we've been using this. So we've been studying cholesterol regulation. As you all probably know, an individual's level of cholesterol in their blood is a primary determinant of their risk of cardiovascular disease. And a lot of work has been done with more classical approaches that have identified this pathway as being critical in regulating the concentration of cholesterol in blood. So cholesterol is very hydrophobic. It doesn't circulate freely. It's packaged into these lipoproteins. The levels of lipoproteins in circulation are controlled by their clearance from the bloodstream. So Brown and Goldstein discovered the LDL receptor that recognizes ApoB on the surface of this LDL lipoprotein. It binds to the lipoprotein and internalizes it into the cell, extracts the cholesterol, and then the LDL receptor goes back to the plasma membrane. So the activity of this pathway is a major determinant of your LDL level. And this is also the pathway that's intervened upon by statins, arguably the most successful cardiovascular medicines that we have. And more recently, over the last decade or two, it's been realized that there's a negative regulator of this process, PCSK9, that is also made in hepatic cells, secreted out into the bloodstream. And what PCSK9 does is it also binds to the LDL receptor, and it diverts the LDL receptor. So rather than coming back to the plasma membrane, PCSK9 induces the degradation of LDL receptor in lysosomes. So it downregulates this pathway and in turn elevates the level of atherogenic lipoproteins. And I think the importance of this pathway to cholesterol regulation is really highlighted by the fact that of all of the known Mendelian causes of very high cholesterol levels, patients who get heart attacks in their 30s and 40s, they all affect genes that are in this pathway. More recently, people have observed genetic variation in a population using GWAS studies, and the Lipids Genetic Consortium, one of the leaders is Kristen Willer here at Michigan. They've looked at hundreds of thousands of patients, and they've looked for spots of the genome where variation is associated with differences in lipid traits. And in this study, they looked at 188,000 individuals, and they found over 150 loci that were associated. But I think a caveat of this work is that for most of these loci, we don't understand the mechanistic association. Why is it that variation there leads to a dysregulation of cholesterol levels? And moreover, even if we look at all the loci that we know about, we can only explain the heritable risk of hypercholesterolemia only about 20 or 30%, suggesting that there are many unknown genes that are impacting cholesterol regulation that have eluded kind of classical genetic approaches. So we came into this area with a very specific question. We were studying PCSK9 secretion, and we had reason to believe that when PCSK9 was secreted from a cell, it wasn't a passive process. So shown here is PCSK9 in the ER, where it's first made, and as proteins enter the secretory pathway, they get incorporated into these membrane-bound vesicles, these spheres that carry them forward in the secretory pathway and out of the cell. And we had reason to believe, like I said, that there was a specific receptor that recognized PCSK9 and recruited it into these vesicles, but we had no idea what it was. So I became very interested in trying to discover this cargo receptor for PCSK9, both because it was interesting for the kind of fundamental cell biology, but also as a clinician, there was an obvious therapeutic relevance here, that if you found an interaction that was important for PCSK9 secretion, if you could disrupt it, you would predict that you would have a lipid-lowering effect and a cardioprotective effect. There were a number of reasons why conventional kind of biochemical approaches might be limited here, and those are underscored by the fact that there are thousands of proteins that are known to be secreted from human cells, but we know of a corresponding ER cargo receptor for only a handful of them. So we wanted to take a new approach. We wanted to use CRISPR, and so we imagined if we took a cell that was secreting PCSK9 into the extracellular space, and we disrupted, using CRISPR, the cargo receptor, that we might expect to see an accumulation of PCSK9 inside the cell and a reduction of PCSK9 outside of the cell. So the challenge here was that we really wouldn't know where to start. So if there are 20,000 genes in the genome or so, how would we go about picking whatever we think the cargo receptor might be? We had really no good clues. But what we were able to do with CRISPR is rather than testing them kind of one at a time and taking decades to try to figure this out, we could test all of them simultaneously. We could introduce mutations throughout the genome. So we used a CRISPR library that had 123,000 guide RNAs in it that targeted virtually every gene in the genome. And moreover, it did it 6 independent times. There were 6 guide RNAs targeting each gene. And so we could use this library to infect these cells, grow up hundreds of millions of cells in a dish, and then each one would have a different mutation and a different coding sequence in the genome. And we could look for those mutants that had a defect in PCSK9 secretion. The challenge is, how do we see it? So to be able to do that, we fused PCSK9 to a fluorescent protein, GFP. So now we would have a basal level of green fluorescence in these cells, and if we disrupted a cargo receptor, we'd be able to see that because it would now fluoresce more brightly. It would have more PCSK9 inside the cell. Importantly, we could not only see it, but we could use a flow cytometer. We could sort 70,000 cells per second, and we could tell the flow cytometer if it meets a certain fluorescence threshold, sort it into one tube. If it doesn't, sort it into another tube. So we could physically separate those mutants, and then we could look for the mutations that were in those mutants that caused the phenotype. As we thought about how to do this, though, we realized that there were probably a lot of ways that you could manipulate a cell and make it generally sick, and you would cause a global defect in protein secretion. It wasn't anything specific to PCSK9, it was just a generalized global protein secretion defect. So to work around this, we built in an internal control. We also co-expressed another secreted protein called alpha-1 antitrypsin, and we fused that to a different fluorescent protein, mCherry. So now we could see the intracellular levels of both proteins simultaneously. And our prediction then is if we had a mutant that was defective for the secretion of proteins just globally, that both the green and the red signals should accumulate in the cell. Whereas if we had a mutant that had a specific defect for one or the other, we could pick that apart because we could see that only one of the fluorescent signals would go up. So our strategy then was to take these cells, again, to infect them with this lentiviral CRISPR library, targeting each gene in the genome 6 times, testing 200 cells for each guide RNA. So we had about 25 million cells that we tested. And then we passaged them for 2 weeks, and then we put them on a flow cytometer. And when we did that, we asked the flow cytometer to do 2 things. First, to only look at the mutants that were normal for alpha-1 antitrypsin mCherry. Again, we don't want to find nonspecific mutants. So within that middle 2/3 of the mCherry signal, we then sorted out on the green channel the brightest cells and the least bright cells. So the ones that had the most PCSK9 stuck in the cell and the ones that had the least. And then we separated those into 2 different tubes, and we did deep sequencing with next-gen DNA sequencing, and we got about 250 million different reads from 210 million sorted cells. And we were able to quantify the abundance of every guide RNA in this tail of the distribution and this tail of the distribution to get a sense of how each guide RNA was affecting PCSK9 secretion. To get a better sense of the signal and the noise here, we also did this 4 times, and we looked for things that were consistent across independent biologic replicates. And when we looked across the entire genome, testing every gene now, we can see an enrichment score. How often is a guide RNA targeting that gene present in the cells with high levels of PCSK9? It was very clear from this screen that we had one dominant hit, and that was SIRF4. And when we looked at the guide RNA level data, we saw that the effect here was consistent across biologic replicates. So this is the mapped reads in the GFP high population versus the GFP low population. These are the 6 different guide RNAs in the library targeting SIRF4. And if we put all 123,000 guide RNAs in a rank order list from most enriched to least enriched, we saw that the top 4 were all targeting SIRF4, and another one was the 8th most enriched. So we had to do follow-up validation and to kind of prove this and validate it. But we were very confident at this point. The likelihood that this happened by chance alone was almost infinitesimal. But we did validate this, so we made individual guide RNAs targeting SIRF4, and we found indeed that these guide RNAs induced a specific accumulation of PCSK9 with no change in alpha-1 antitrypsin. And this was true for several different guide RNAs that we tested. So I'm going to summarize our follow-up work, our cell biology and biochemistry that we did just for the sake of time. But we went on to show that when we deleted SIRF4, in fact, we did have a reduction in secretion of PCSK9. We did have an accumulation of PCSK9 in the endoplasmic reticulum, so this really did seem to be a defect in ER exit. We found that SIRF4 was localized to the ER membrane here, so it was in the right place to be acting as the cargo receptor. And we also found that these two proteins physically interacted with each other. And we tested the 21 most next enriched candidates from the screen, and none of them were true positive. So we showed that this was really the primary determinant in these cells of specific PCSK9 secretion. So at this point, we feel pretty confident that in these cells, SIRF4 is acting as a cargo receptor, promoting the secretion of PCSK9. But so far, everything that I've talked about or showed you has all been done in immortalized cells growing in a dish. And we really don't know how relevant that is to the physiologic environment. And so we wanted to test that, and so we again used CRISPR. And because of CRISPR, we were able to do this fairly quickly. So in a mouse, so now instead of injecting CRISPR reagents into cells, we're injecting them into fertilized oocytes of a mouse, and we're transferring those into a pseudopregnant mother, and we're getting pups that are born from that litter. And we can see that when we inject these CRISPR reagents targeting SIRF4, that we indeed got several different alleles with germline transmission where we had disruptions in SIRF4 function. So we were able to make a knockout mouse for SIRF4. So what we wanted to do is to make a complete knockout. So the first generation just has one deleted copy of SIRF4 and one normal copy of SIRF4. What we really wanted to do was completely knock out SIRF4 from the mouse and see what happened to cholesterol. So we set up intercrosses you would expect by Mendelian ratios that a quarter of the progeny from these heterozygous mice should be knockout mice. But what we found is when we looked at the time of weaning, that all of these mice either were heterozygous or they were wild type. None of them had the knockout phenotype. So this indicates that the knockout mice are dying sometime during embryonic development. We were able to do time matings, and we were able to see that if we looked at the blastocyst stage at E3.5, that there were knockout blastocysts. But if we look later at E9.5, they were dying somewhere in that window in between. So this indicates that SIRF4 has a critical role during early embryonic development. We don't really understand what that might be, but we do know that PCSK9 is perfectly dispensable for embryonic development, so it's not an effect on PCSK9, and it suggests that there are other functions or other cargoes for SIRF4 that are important during embryonic development. It also prevented us from testing the role of SIRF4 in cholesterol regulation because we couldn't generate the knockout mice. So to circumvent this, we also used CRISPR again. And in this time, we allowed the mice to go through embryonic development. Normal kind of wild type mice, they have plenty of SIRF4. And then once they were adult mice, we could target SIRF4 just in the liver, just in the tissue where PCSK9 is being made. So we could use viruses that deliver guide RNAs targeting SIRF4. We did this in collaboration with a previous postdoc in David's lab. And when we looked, so after injection of SIRF4 guide RNAs, we looked at the PCSK9 levels in circulation. LAC-Z is a negative control. It shouldn't cut anything. And then PCSK9 is a positive control to disrupt PCSK9. What we found, in fact, was that we saw about a 50% reduction in PCSK9 levels in circulation, kind of similar to the quantitative effect that we saw in cultured cells, and consistent with the idea that this SIRF4 role in PCSK9 secretion was not just an artifact of our immortalized cells in a dish, but actually physiologically relevant too. What was really surprising, however, was when we looked at cholesterol levels. So we looked at these same mice that had been targeted for SIRF4, and we saw that 2 weeks after injection, the cholesterol levels were reduced by about 75% or so. But if we looked at 4 weeks or 8 weeks after injection, the cholesterol levels were almost undetectable. There's greater than 95% reduction in the cholesterol levels. This was very shocking, and the reason for that is we have other models of PCSK9 deficiency that don't show nearly the same magnitude of cholesterol lowering. So the PCSK9 knockout mouse itself, or another mouse that has a defect in PCSK9 secretion, the cholesterol reductions that are seen are on the order of 40 or 50%. What we're seeing is a greater than 95% reduction that we can explain with a defect in PCSK9 secretion alone. This suggests that SIRF4 is doing other things that are important in cholesterol regulation. We got a clue as to what that might be from proteomics. So we took liver-derived cells growing in a dish. We knocked out SIRF4. We looked at everything that they secreted into the media above them, into the conditioned media. And we did quantitative proteomics in collaboration with the core here. And we quantified everything, and we looked at which proteins became less abundant, and they were secreted less when you knocked out SIRF4. And surprisingly, we found that the 2 most depleted proteins were apolipoproteins, which you might remember are part of these lipoproteins that circulate cholesterol. And so they're on the surface of these lipoproteins. And this suggests that SIRF4 is not only trafficking PCSK9 out of the cell, but also ApoB. And so we looked at those mice that were knocked out for SIRF4, and we found indeed that ApoB was greatly reduced in circulation. And if we looked in the liver, it wasn't just that these mice were making less ApoB, they actually had an accumulation. They just couldn't secrete it very well. They were able to make albumin just fine and other kind of abundant hepatic proteins that are secreted into circulation. This wasn't a generalized defect of protein secretion, but had some specificity for ApoB and for PCSK9. We also went back and looked at our heterozygous mice that have just 1 copy of SIRF4, and we found if we looked at the liver, there was actually an accumulation about 3-fold or 2-fold of ApoB in the liver of those mice with just a partial reduction of SIRF4, whereas PCSK9 was unaffected. So it seems like ApoB is not only dependent on SIRF4, it seems to be more sensitive than PCSK9 to SIRF4 depletion, whether that's partial depletion or complete depletion. So we also want to confirm this with a genetic model. So the previous experiments were with a viral delivery system. We have worked on making a conditional allele where we can knock out SIRF4 only in specific tissues, just in the liver. And this is preliminary data, but this is consistent with what we saw with the viral delivery system. Again, when we knock out SIRF4 only in the liver, embryonic development is fine. The mice look normal, but their cholesterols are markedly reduced, over 90 or 95% reduced. So this is all very interesting and exciting. It suggests that targeting the cargo receptor for SIRF4 may have an even more profound lipid-lowering effect than just targeting PCSK9 by itself, because we might also disrupt apolipoprotein B secretion. So it's interesting and exciting, but we really don't understand why this is happening. We have a model for the structure of SIRF4 where it has 5 different transmembrane domains, and some of these domains reach into the cytoplasm and interact with the vesicle machinery. But cargo recruitment is thought to occur through these luminal domains. So this reaches into the ER lumen, recognizes cargoes, and pulls them into these vesicles. So how is it that this is happening? Are PCSK9 and ApoB binding to the same cargo binding domain? Are there distinct domains? For other cargoes, do they use the same domains? We really don't know. And this is important to figure out because if we were to design an inhibitor to try to disrupt these interactions, if they were on distinct domains, you might be able to come up with a specific inhibitor to affect some, but not others. So we have a good fluorescent readout, again, for PCSK9 secretion. So these cells make fluorescent PCSK9, and when we knock out SIRF4, they accumulate more of it because they can't secrete it well. Now we can add back SIRF4, and it normalizes that level of PCSK9 inside the cell. So now we can test different mutant copies of SIRF4. We can add them back and see whether they shift fluorescence or not as a kind of rapid and quantitative readout of SIRF4 activity. So the first thing we tried to do was to take this protein and to chop off different domains, kind of classical biochemical experiments here, where we truncated different domains and we saw whether or not they could still rescue secretion. And we found that it wasn't so simple, that this was too blunt of an instrument, that if we deleted any of these domains, that the protein lost its function. It was no longer able to support PCSK9 secretion. So we needed a more kind of fine-tuned, precise way to to change SIRF4. And so we turned to error-prone PCR. So most of the time when you think of PCR, you think you want to copy a gene sequence. You want to do that in a you want to have high fidelity in doing that. You don't want to introduce mutations. But we can use polymerases that are engineered to be more error-prone so that when we amplify this, we can get a library of mutants that have random changes in the coding sequence, throughout the coding sequence. And we can do this at scale again. So we made 8 million different mutants, saturating every possible nucleotide change that there could be in SIRF4. On average, they had 1.3 mutations per copy of SIRF4. So now we were able to go back into our reporter cells and to say, if we have normal PCSK9 accumulation inside the cell here, if we add back a functional SIRF4 copy, we expect, again, we're going to rescue that effect. We're going to reduce the fluorescence inside the cell. Whereas if we add a copy of SIRF4 that's non-functional, it's not going to shift the fluorescence. It's going to look just like the mutant cell. If we take this library of mutants now, all possible coding substitutions, we would expect that we would see kind of a bimodal distribution, that some of these mutants might be functional and some might not be functional. And now we can use flow cytometry and we can sort out the tails of the distribution here and do next-gen sequencing again and get an idea of every possible mutation. Does it disrupt function of SIRF4 or does it not? Is it a tolerated mutation? So we did this across the coding sequence of SIRF4, like I said, and here you see a display for every possible substitution in the SIRF4 coding sequence. How much of an impact does it have on SIRF4 function? So the higher it is on this y-axis, the more disruptive this mutation is. And this was striking to us. So if we look, where might a PCSK9 binding domain be? You might predict that if we hit the area of SIRF4 that binds to PCSK9, it shouldn't tolerate mutations very well. And if we look at the first luminal domain, we see that all of these mutations seem to be well tolerated. We don't really disrupt the function of SIRF4 introducing mutations here. If we look at the last luminal domain down here, we don't disrupt function again. But if we look in this middle luminal domain, even in the first half, there's little effect. But over the last 30 or 40 amino acids or so, all of these mutations seem to be disrupting the ability of SIRF4 to promote PCSK9 secretion. So I think this suggests to us, we're in the process of testing this, that the PCSK9 binding domain is in this region. And if we can show that and we can reconstitute it, now we can try to purify these proteins and get them to bind to each other in an in vitro environment. We can't do that with a full-length protein because it has so many transmembrane domains. It's very hydrophobic, and we'd have to use detergents to solubilize it, and that could break apart protein-protein interactions. If we can reconstitute this with a smaller domain, now we can study the kind of in vitro binding affinity, the variables that affect binding, and we could even screen for small molecules that might disrupt that interaction. The other thing we can do is we can ask whether or not these mutants which disrupt PCSK9 secretion, whether they also disrupt the secretion of ApoB or other SIRF4 cargos, to get at this idea of whether or not the cargo binding domain of SIRF4 is unique or if it's separate for the various cargos. So there's a lot that we're still following up on for SIRF4 biology and its role in cholesterol regulation. I just want to switch gears for the last bit of my talk here and tell you a little bit more about another screen that we did that's more preliminary but very exciting. And that's looking for modifiers of Ldl uptake. So this is that kind of first pathway I showed you where cells will take low-density lipoprotein in the extracellular space, and through the LDL receptor, they'll bind to it and they'll internalize it into the cell. And as we were setting up these assays, we realized that this would be very easy to do a genome scale CRISPR screen on, that we could mutagenize cells, and if mutants had disruptions in LDL uptake, like knocking out LDL receptor, they should take up less LDL, and you can see that on a flow cytometer because they'll be less bright. Whereas if you have a mutation that enhances LDL uptake, then you'll see more fluorescence associated with the cell. This is also an appealing screen because we have a lot of built-in controls. We know some of the players that are involved in this process. We can look to see whether or not our screen identifies them, but we can also look for novel things too. So we used a hepatoma cell line, an HUH7 cell, and we showed that indeed they would take up this fluorescent LDL, and that if we knocked out the LDL receptor, it would disrupt that uptake. So they were using this pathway for LDL uptake. And if we starved the cells of cholesterol, we grew them under conditions of low lipoprotein concentration, that they would actually take up more. They had upregulation of this pathway. And again, blocking LDL receptor disrupted this uptake. It was an LDLR-dependent mechanism. So our screen was pretty simple then. We took these cells, these hepatoma cells, we infected them with the same guide RNA library of 123,000 guides, and then we split the cells. We either grew them under lipoprotein-rich or lipoprotein-depleted conditions. We applied fluorescent LDL, and then the mutants took up more or less LDL depending on the mutations that they had. And we again used flow cytometry to sort out the mutants that took up the most LDL and the ones that took up the least LDL. And so the first thing we do is we look across the genome at what do we expect to find? Did the screen work? And so it's reassuring to us when we see one of the top hits is LDL receptor itself. And there's also other positive controls that are involved in the expression of LDL receptor in the SREBP pathway, and those were identified too. When we looked under lipoprotein-rich conditions or under lipoprotein-depleted conditions, we identified these genes. The false discovery rates are shown with dashed lines, 1% and 10% here. There are several other hits that we're in the process of exploring. When we look the other way, we look at genes when we mutate them that we enhance LDL uptake. There aren't as many controls here. MyLip or IDLE is really the only one that we had. And it was not giving it as strong of a signal, but it was still among the top 10 or 20 genes or so. And so that was reassuring. And then we also looked at what were the other things that we found, what were the other hits from the screen, and you can see that there was a lot of concordance. The genes that rose to the top of the list for one arm of the screen also rose to the top of the list for the other arm of the screen. If this was all just kind of random variation, you would expect that they would be completely distinct. So I think together, the positive controls being identified and the concordance between the two arms of the screen really makes us think that what we're seeing is true biology here. But of course, we didn't do this just to identify the known regulators. We wanted to find new things that were involved in LDL uptake. And we started going through these genes and trying to make sense of what we found. And it was striking that there was genes involved in the same pathway that we were seeing again and again. So the exocyst here is a multi-protein complex, and we kept seeing components, XOC7, 3, 4, and 8. And then these other genes that are not important for you to know other than to know that they interact with the exocyst. They're in the same pathway. And we saw this in both arms of the screen again. And we also saw for mutants that had enhanced uptake, we saw a negative regulator of this pathway on both arms of the screen. And so what is this pathway? I don't want to belabor the insulin signaling pathway because it's very complicated. But all you really need to know is that the way insulin triggers glucose to go into cells in large part is dictated by its ability to take glucose transporters that are normally not at the cell surface, but when insulin binds to the insulin receptor, it sets off this signaling cascade, and the end result of that is these vesicles mobilize to the plasma membrane. So now the glucose transporter is out here at the periphery, and it allows glucose to go into cells. So there's a lot of work that's been done over many, many years by many labs identifying all of the different components of this pathway. This is just a handful of them. But we kept seeing that all of these genes that were involved in this process were also hits on our screen. And so that's very interesting, and it suggests that the machinery that the cell uses to get glucose transporters to the cell periphery upon insulin stimulation is perhaps also involved in this recycling of LDL receptor. There's not a whole lot that's been known about the molecular machinery that brings LDL receptor back to the cell surface. We went and we tested this. So we used single guide RNAs targeting either the LDL receptor itself or exocyst components, and we found indeed when we knocked out these genes that we disrupted LDL uptake. This did not seem to be a non-specific generalized effect because when we looked at uptake of other molecules like dextran, which is a fluid phase marker, or transferrin, there was no defect in their ability to take up those molecules. In fact, for transferrin, if anything, there's a slightly enhanced uptake of transferrin. So this was not a global endocytosis defect. It seemed to be specific to the LDL receptor. And it turns out that one of the world's experts on insulin signaling was right across the hall from us when I joined the lab, Alan Saltiel, the former director of the LSI, who's now at UC San Diego. So we shared this data with him, and he became very excited about it, and we're collaborating on this. And they did an experiment where they took primary mouse hepatocytes, and they stained for LDL receptor. And they found under basal conditions, there was actually a surprising amount of LDL receptor inside the cell, not at the plasma membrane like you might expect. But that when they treated these cells with insulin, that there was this translocation of LDL receptor from the inside of the cell to the periphery of the cell, kind of consistent with what they see with the glucose transporter. And they have many different small molecule compounds where they can disrupt insulin signaling. And so, again, they see here that there's intracellular LDL receptor, and then there's a mobilization with insulin treatment to the periphery of the cell. But when they block insulin signaling with these various inhibitors, it prevents this translocation of LDL receptor to the cell surface. So this is exciting. It's still somewhat preliminary, but it suggests to us that indeed this pathway of insulin signaling that results in mobilization of glucose transporter is also somehow involved in the recycling of LDL receptor to the cell surface. And I think there are important clinical implications of this. I think it's well established that diabetes is a risk factor for cardiovascular disease. We've known that at least since the Framingham study. And I think the mechanism of this is not entirely well understood. But when I was in residency here, it was around the time that the lipid guidelines were changing. And rather than focusing on an LDL target level and trying to bring patients down to a goal, we were focused more on the risk of cardiovascular disease. And if you looked at the algorithm of whether or not you should put a patient on a statin, if they had diabetes, that alone, regardless of their LDL, almost warranted them being treated with a statin just because their cardiovascular disease risk was that much higher. And I think despite this, like I mentioned, I think we don't have a great understanding of why diabetes leads to higher rates of cardiovascular disease or why insulin resistance in the context of the metabolic syndrome is associated with lipid abnormalities. I think that our data suggests that perhaps part of this is that if you have resistance to this insulin signaling pathway, in the same way that you don't translocate the glucose transporter, maybe you don't also translate the LDL receptor, leading to lipid abnormalities. Another important consideration of this is if you think of different therapies, different diabetes medications, some of them have been shown to lower the risk of cardiovascular events, some have not. It's not entirely clear to me, at least, why that is. I think that this suggests maybe if you had treatments that were sensitizing, that sensitize the cells to insulin signaling, perhaps you might have a more beneficial effect on LDL receptor expression at the cell surface and more of a cardioprotective effect. But that's all speculative, and we've got more work to do on that. So in conclusion, I think we feel like we've convinced ourselves at least that the cargo receptor SIRF4 has this important role in PCSK9 and apolipoprotein B secretion, and through those dual roles, it plays a critical role in modulating cholesterol homeostasis. I think we also have preliminary evidence that suggests this kind of metabolic crosstalk between insulin signaling with both glucose transport and lipoprotein metabolism. And I think finally, these experiments are also kind of a proof of concept that this technology is very powerful. It's an unbiased and high throughput way to query the genome and to find these novel genetic interactions. And as someone who's kind of starting my own research program, this is very exciting to me and a focus of what I want to do to leverage these high throughput and unbiased technologies to find new genetic interactions that are important in health and disease. And so with that, I thank you all for your attention. I just want to thank my collaborators, the other members of the Ginsberg Lab, my funding sources and mentoring committee, and especially the support of my department and division, who've been incredibly supportive of me as I've been going through this phase of my career. So thank you all, and I would be happy to take any questions.
Ryan, really very, very impressive, wonderful work. Let's take some questions. Jim.
Yeah, I would second that. When you lower SIRF4 cholesterol levels, what happens at a cellular level? Endogenous cholesterol metabolism production.
Sure.
And is the cellular cholesterol lysosomal cholesterol?
Yeah, great questions. The short answer is we don't know. We're starting to characterize those mice in more detail. I think having this conditional allele with a liver-specific knockout will allow us to kind of rapidly generate more mice where we can do some of those kind of studies. I would say what we do know about kind of the function of the hepatocytes is that morphologically, looking at tissue sections, there don't seem to be any gross abnormalities that we see. And if we look at like synthetic function of hepatocytes, if we look at other abundant secreted proteins, there doesn't seem to be a big change. But as far as kind of more nuanced phenotyping of the cholesterol inside the liver, we don't have a great sense of that yet.
Other questions?
Two questions. First is, my guess is that PCSK9 levels have been measured in some cohorts with genetics. Have you looked at whether SIRF4 shows up in Mendelian randomization, for example?
Not in Mendelian randomization specifically. We have looked at the GWAS data sets to see whether or not there might be a signal associated with SIRF4 for different lipid traits, for example. The evidence isn't clear. So there is one SNP in an intron of SIRF4 that is associated with hyperlipidemia, but it's also in linkage with some other SNPs that have an association, and whether it's kind of truly an independent variant that has an association, we don't know yet. I will say that we do know from our mouse genetic data that we would not expect that one copy being affected. So like in a GWAS study where you might pick up common variants that affect one of the two alleles, our animal model data and our cell culture data really suggest that complete deletion of SIRF4 has a big impact, but that partial, one copy, functional copy, one non-functional copy does not. So I'm not sure that we would expect to see anything in a GWAS anyway, but there's some evidence that perhaps there's one intronic SNP that might be associated.
Cool. One other question. You talked about crosstalk between the insulin GLUT4 pathway and also LDL receptors. If that's the case, why don't we see variation in LDLs very much between fasting and fed states in humans?
That's a great point. Very good question. I don't know. I think we're just starting to explore the relationship of this, insulin signaling pathway and LDL receptor translocation, and I don't have a great answer for that, but it's certainly something we're interested in.
Yeah, good.
What about mutations in Escort polymorphisms and the Escort pathway and hyperlipidemia? Did
Sure.
they come up in the screens that
Yeah.
Miller did?
Just to kind of keep this focused, I didn't spend much time talking about the other. Oh, yeah. So for our LDL uptake screen, there were potentially dozens of hits here. And I focused on the ones where we had more of a sense of the biology to explain, potentially how this was mediating the effect. The Escort proteins, there were a couple that were involved, or a couple that we saw here that had a significant signal that we have not yet validated or done any follow-up studies on, but it did seem like we identified some of the Escort machinery as being involved in LDL uptake.
Great. If not, let's just thank Brian one more time for really exciting.